Hello, my name is Isabel Reid, and I'm a recent graduate from the University of Michigan in Ann Arbor. This summer, I got to work in the Fitnani lab through the NIH SenNet CUSP internship program at Columbia University Irving Medical Center. On this slide, you can see a brief overview of what the Fitnani lab does. This consists of where we get our tissues from, the University of Edinburgh and the New York Brain Bank at Columbia. From these areas, we get brain regions of the dorsolateral prefrontal cortex, Brockman areas 9, 46, and 44, the posterior hippocampus, along with the spinal cord lumbar. All of these regions control executive function, emotions, memories, and movement, all of which pertain to the disease of ALS. Next, our tissues go to the biological analysis core. In this area, there are a few different aims that we are set to meet. The first one being 10x Visium spatial transcriptomics, second being single nuke RNA-seq, the third is 4I with an antibody panel, and the fourth is validation and imaging. Finally, the fifth is modeling and perturbations. During my internship, I mostly focused on the first aim of 10x Visium and spatial transcriptomics. Next, we go to the data analysis core, which I personally was not very involved in, but this mostly takes the biological data that we learned and the spatial transcriptomics, puts them together so that we can figure out what conclusions can be drawn from the overall research. As I said before, I mostly focused in the 10x Visium spatial transcriptomics data. Within this, first we must do tissue quality control of the different samples that we receive from both Edinburgh and New York Brain Bank. We do methanol fixation and H&E staining to then visualize the tissue with bright field imaging and see if we want to keep the sample for downstream analysis or drop the sample. This is depending on whether there's freeze fracture, whether the desired anatomy is clearly visible, and many other factors. From here, if we do choose to keep the sample, it either goes through the tissue optimization protocol or library construction. During the summer, I did an experiment with the tissue optimization protocol. My main goal was to see whether the tissues between the different brain banks produced different results. For the U54 project that my mentor was working on, she has processed a total of 550 samples. Of these, some of them come from the Edinburgh Brain Bank and some of them come from the New York Brain Bank. I wanted to see if there is a difference between the outputs between these two locations. In order to do this, I did two different experiments. The first one is that I determined the integrity of the RNA of the different samples from various sites by determining the RIN. The second is to determine whether the tissue permeabilization times in the tissue optimization experiment varied for the different tissues between the sites. During the Visium pipeline, we get various outputs that I'll explain here and reference later on in the slides. As you can see, there's a slide here, or an array of a Visium slide, which contains tissue. The different outputs measures various different things, including the overall number of reads per array or square on the Visium plate, which you can see here, along with the number of spots that the Visium slide that are actually covered by the tissue. You can see here that on this specific array, it is almost completely covered by tissue. It also measures the median number of genes that existed on each individual spot that you can see within the square. And finally, the number of unique molecular identifiers that exist per spot. I compared these outputs between the different sites to determine whether or not there is an effect of the tissue site on the outputs. You can see here a graph of the average New York Brain Bank versus University of Edinburgh output, and you can see that the data seems to be fairly similar. Along with this, it's important to note that all of the data meets or is very close to meeting the benchmark the benchmark values that we as a lab have set. These exist to be 170 million reads per array, 40,000 reads under each tissue per spot. And for the dorsolateral prefrontal cortex, we want 700 median genes per spot, while 200 median genes per spot is the benchmark for the hippocampus tissues. You can see on this graph that all of these benchmark values are met with the exception of the dorsolateral prefrontal cortex reads under tissue per spot for University of Edinburgh, as well as the median genes per spot for the frontal cortex tissue from the New York Brain Bank. However, both of these values are extremely close to the benchmark value. From here, I selected a cohort to represent the overall populations from the different brain banks with the specific tissue type. Therefore, there were 5 different tissues: prefrontal cortex from both Edinburgh and New York Brain Bank, hippocampus from both sites, and finally, spinal cord from only the New York Brain Bank, as we did not receive any from the Edinburgh tissue. You can see the demographics of these tissues chosen depicted in the figure here. These samples were chosen to represent their population based on which samples most closely related to the average values of that population. So you can see here the comparison between the specific chosen samples for each site versus the population. Here we see the tissues chosen from the University of Edinburgh. On the left is the frontal cortex, and on the right is the hippocampus. We see that the tissues that are chosen are extremely close to the values of the average. We can see here that the same is true for the New York Brain Bank chosen tissues versus the population. Next, I did a quality control analysis of all of the tissues that I chose to ensure that they didn't have substantial freeze fracture and that they had the desired anatomical regions. These next few slides I'll go through to depict that there isn't any freeze fracture and that the anatomical regions are shown. As you can see in the hippocampus, this one has the dentate, and so does the Edinburgh tissue. And finally, the spinal cord lumbar has both horns that we want to see. Next, I went through and actually calculated the RIN for all of these tissues, and you can see from these results that the New York Brain Bank tissues did have lower RIN than the Edinburgh tissue. However, this begs the question of if this has an actual effect on our results. Therefore, I compared the RIN to the median genes per spot that were found in the spatial transcriptomics analysis. I found that there didn't seem to be a real correlation between them. While it may seem that there's a positive correlation in this graph, due to the existence of the outliers and the small sample population, no real conclusions can be drawn. This is also true when I compared the UMI per spot versus the RIN and found that the RIN didn't seem to have any real correlation with the outputs that we had. Therefore, the difference in the RIN between the sites seems to be insignificant for the purpose of our results. Next, I went through and did the tissue optimization protocol by 10x Genomics. For this portion of the experiment, I unfortunately had to replace a few of the samples due to the usage of the entire tissue in the previous experiment. Therefore, I had to replace the frontal cortex for the University of Edinburgh along with the hippocampus for the New York Brain Bank. So again, we'll go through the comparison between the chosen sample and the population and see the similarities between them. Here we can see the sample versus population for Edinburgh and the similarities. And finally, we see that the chosen tissues for NYBB are very representative of the overall population. Again, I went through and did quality control analysis for these new tissues, specifically looking at the Edinburgh tissue and the NYB hippocampus. We see that these again pass the quality control and are able to be analyzed as a representation of the entire population. During the tissue optimization experiment, I used 10x Genomics protocol to determine the specific appropriate tissue permeabilization time for each tissue. Step 1 of the protocol was to do permeabilization and cDNA synthesis. This consisted of having 10-micron sections of the tissue in each 8 array on the slide and performing tissue permeabilization, or allowing the membrane to be permeable, on all 8 tissues at different times. From here, we allowed the cDNA to synthesize and fluoresce. Therefore, it would be visible on the slide with Cy3 imaging. This left us with both the tissue and the fluorescent cDNA. From here, the next step in the protocol was to remove the tissue, the fluorescent cDNA, to determine the ideal permeabilization time for each tissue. From here, you can see an example of the results of the TO experiment from 10x Genomics. On the left, you see the H&E staining, and on the right, you see the Cy3 imaging with the cDNA present. From this experiment, 10x Genomics determined that 12 minutes was the ideal permeabilization time, as it contained both the brightest and crispest image. Here's another example which depicts the importance of the crisp image. Although 30 minutes may look as if it's the brightest signal at first glance, upon closer examination, we can see that 18 minutes contains a much crisper picture of the cDNA, and therefore was chosen as the permeabilization time. I went through this process with all of my tissues. First, the Edinburgh frontal cortex tissue, I personally did not do the experiment as it was already performed in 2020, and due to lack of resources, we decided to use these results in the experiment. Next, for the frontal cortex from NYBB, I determined that the permeabilization time that was ideal was also 12 minutes. For the NYBB hippocampus, I looked at the different anatomical features that seem to have the highest UMI count and looked at where the fluorescent cDNA was both the brightest and crispest in these areas, and determined a permeabilization time of 15 minutes. For the Edinburgh hippocampus, I did the same, and looking at the results a little, we can see that this one was a little bit more tricky. Upon first glance, I thought that 18, 16, and 15 minutes seemed to have pretty bright, crisp images. Upon comparison, looking specifically at the areas where the UMI per spot count was very high, for example in the dentate, we can see that 18 and 16 seem to have much brighter images than 15. When I zoomed in and really looked at these important regions, I saw that 16 minutes, although it seems to be a little bit dimmer, is much crisper. Therefore, I determined that 16 minutes was the ideal tissue permeabilization time. Finally, I determined that the tissue permeabilization time for the spinal cord lumbar tissue from the New York Brain Bank was 10 minutes. In this experiment, to determine whether there was a difference between the two sites, we wanted to compare the results to the originally predetermined permeabilization times set by the lab. These were 12 minutes for frontal cortex, 15 minutes for hippocampus, and 12 minutes for spinal cord. We found that for both sites, these times matched for the frontal cortex. For hippocampus, the New York Brain Bank tissue did have the expected time of 15 minutes. However, University of Edinburgh was slightly longer at 16 minutes. Finally, the spinal cord lumbar tissue had a slightly shorter permeabilization time than expected of 10 minutes. These results suggest that although there may be slight discrepancies between the tissues of the two sites, we cannot determine whether or not this affects the data. This is because when we did the RIN analysis originally, although we found a difference in the RIN, we couldn't determine that these results correlate to the outputs. Finally, with the difference in the permeabilization times, although there is discrepancies, these are very small and there are still fluorescent cDNAs at the expected permeabilization times. In closing, I wanted to add a special thank you to all of the people who helped me over the summer. First of all, Dr. Himali Fatnani was the principal investigator of my lab and played a great role in mentoring and helping me to find my interests within the lab. Shanice Grier was my personal mentor and taught me so many different techniques and helped me to understand the overall workings of the lab. Finally, Nadia Prop was the lab manager and helped me to feel like I belong within the environment. Additionally, I wanted to say another thank you to the Fatnani Lab as a whole, along with the NIH SenNet CUSP internship program, who made this summer possible for me. Thank you.